The research project focuses on Medicago sativa, commonly known as alfalfa. Alfalfa is an important forage crop used for animal feed. Climate change is causing an increase in the frequency of droughts. The research aims to investigate the impact of drought timing on early-stage alfalfa growth. Understanding this impact is crucial for sustainable agriculture. The study will help develop strategies to mitigate the effects of drought on alfalfa production. The experiment investigates the impact of drought timing on alfalfa plant biomass. Key elements include a drought simulation, control plant groups, and drought-stressed groups. Plant groups were subjected to drought conditions at different times, week 2 and week 3. Biomass was measured to assess the effects of drought on plant growth. Surprisingly, drought timing had minimal impact on early biomass within the 1-week drought period. Early season droughts have been shown to significantly reduce plant growth in previous studies. This study investigates whether early season drought impacts the earliest stages of alfalfa growth. The key question is if alfalfa seedlings can withstand short droughts by utilizing stored seed resources. Researchers are exploring the drought tolerance mechanisms of alfalfa seedlings during initial growth phases. The central hypothesis posits that alfalfa biomass will be less negatively impacted by later droughts. This suggests alfalfa plants develop increased drought resilience over time. Early droughts are predicted to have a more significant negative effect on biomass production. Visual aids reinforce the concept of varying plant group responses to drought timing. The experiment investigates the impact of drought on alfalfa plant growth. 3 treatment groups were established: a control group, a 2-week drought group, and a 3-week drought group. Each treatment group consisted of 14 pots, with each pot containing 1 alfalfa plant. All plants were grown from the same seed package to ensure genetic consistency. Total dry mass was measured for each of the 84 individual plants to assess the effect of drought. Alfalfa seeds were planted and grown in controlled pot conditions. All plants were exposed to UV lamps for 12 hours daily to simulate sunlight. Control plants received 300 milliliters of water twice weekly, while drought-stressed plants underwent weekly drought periods. The experiment lasted 4 weeks to observe the effects of drought on alfalfa growth. One-way ANOVA was employed to analyze the experimental results. The analysis focused on comparing the final dry masses of different experimental groups. The experimental design included a control group with standard conditions. A week 2 drought group experienced drought conditions during the 2nd week. A week 3 drought group experienced drought conditions during the 3rd week. One-way ANOVA facilitated statistical comparison between these groups to determine significance. A bar graph illustrates the mean dry mass of alfalfa plants under different drought conditions. The experiment included a control group, a week 2 drought group, and a week 3 drought group. The week 2 drought group exhibited the highest mean dry mass in the experiment. The control group showed a mean dry mass lower than the week 2 drought group. The week 3 drought group recorded the lowest mean dry mass among the 3 groups. A high p-value, 0.8138, indicates that the observed differences in mean dry mass were not statistically significant. The researchers' hypothesis regarding drought timing was not supported by the data. Drought timing did not significantly influence early biomass production in the alfalfa field. This suggests that the timing of drought stress may not be a primary factor in early growth stages. Other factors likely had a more substantial impact on early biomass development. These factors may include variations in light exposure, nutrient availability, or seed vigor. Small differences in these environmental or biological factors could outweigh the effect of drought timing. Young alfalfa seedlings may initially depend on stored seed reserves. These reserves provide essential nutrients and energy for early growth. Seed reserves can enhance drought survival during the seedling stage. Experimental drought conditions may not have been severe enough to significantly impact seedling development. Early season drought typically impacts mature plants more severely. This study investigates the drought tolerance of very early alfalfa seedlings. Initial findings suggest that young seedlings may exhibit unexpected drought resilience. Future research could involve extending the drought period or analyzing root-to-shoot ratios.